see us now? I think there's 60 participants here.
Yeah, we are also now live on YouTube.
Okay, I guess so.
Yeah. So people can ask questions also on Slack, and then these questions will be transferred to the Q&A. So we only have to watch on the Q&A, and you could also read the questions on the Q&A if you want, but Crasher will read them aloud to you after your talk.
Yeah.
You can see my pointer, right?
Yes, yes.
Okay.
Yes. Mm-hmm. So the Q&A questions will be upvoted, and if there's too many questions for the 5 minutes, I will just go through the top questions.
Sounds good.
Yeah, a few more minutes. Yeah, well, a few more seconds.
A few more seconds, yeah.
Yeah. I'll start at 8 exactly.
So Krisho, you will give me the 5, right?
I'll waive 5.
Okay, because I will only have your video on top. I mean, I'm paying attention to time, but just in case.
Okay, sounds good.
Carried away. Okay, great.
All right, I think we're here. Yeah. Good. All right. So welcome to the one of the parallel sessions. So this is going to be the session on dynamics of structured neural networks. And our first speaker is Juliana Georgieva from the Max Planck Institute for Brain Research and also at the Technical University in Munich. And she's going to talk about spontaneous emergence of structure and recurrent networks with a triplet STDP rule. Okay, Juliana, the talks are going to be 25 minutes, plus 5 minutes of questions. If you have a question, please type it in the Q&A box. And if you think that a question is particularly valuable, also give it a thumbs up and it will move up. So the best questions will be the ones that we're going to ask in the Q&A period. Juliana, go ahead, please.
Okay, great. Thank you very much to Krisho and to Thilo for inviting me to take part in this session and in this conference. So what I'm very excited to tell you about today is some recent work that we have done on understanding how non-random structure emerges in recurrently connected networks from a particular spike timing dependent plasticity rule that depends on triplet interactions of spikes. So we know that Hebbian plasticity is one of the best known activity dependent mechanisms that connect cells that fire together. So we know that the persistent firing of neuron A, if that drives the persistent firing of neuron B, then the connection between A and B will be potentiated. And this kind of Hebbian plasticity has been sort of argued to be one of the key mechanisms for the emergence of cell assemblies that have also been termed Hebbian assemblies. So these are groups of neurons that are strongly connected to each other. And they become strongly connected to each other because they're repeatedly co-activated. So they experience correlated activity and they become strongly connected. And these cell assemblies are relevant because when they're present in recurrent networks, such as the ones I'm showing here on the slide, they generate very different types of dynamics than these networks that just have uniform and random connectivity throughout. So here we see the activity of such a uniformly connected network at the top, which is asynchronous and irregular. And at the bottom, we see the activity of these structured networks that have these cell assemblies in them with higher probability of connection within an assembly or stronger connection weight. So these have been suggested to be the building blocks of cortical microcircuits and to be recruited in the execution of different types of functions. And what is interesting about them is that they enable the cortex to generate these reverberating patterns of non-random activity. These have been implicated to be responsible for the representation of different stimulus features. For example, different tastes in gustatory cortex, and more prominently in the visual cortex, various studies have shown experimentally that such cell assemblies indeed exist. So here I just show an example from the work of Tomašić-Flogel, where they measured specifically connection probability for neurons as a function of their similarity of stimulus preference. So these are simple cells in the visual cortex that are tuned to different orientations, as is shown here in the schematic. And if one looks at the probability of connection, you see that neurons that have similar stimulus preference are more likely to be connected than neurons that have orthogonal stimulus preference. And what is interesting is that this type of pattern of connectivity, presumably giving rise to these self-assemblies, emerges after eye opening. So in the mouse, eye openings around P15. And so it has been proposed that potentially this structured visual input coming through the eyes after eye opening could give rise to the formation of these cell assemblies. Now, cell assemblies have also been seen in other kinds of brains and brain circuits. So here we see an example of the optic tectum in the zebrafish. This is functional imaging, so 2-photon calcium imaging at different ages. So DPF stands for days post-fertilization. What is important here is the emergence of these beautiful connectivity structures. So this is all functional connectivity, so functional cell assemblies in these in the optic tecta of these animals. But unlike the visual cortex example that I showed you on the previous slide, here it seems that these types of cell assemblies emerge completely in the absence of patterned external input. So you can block visual input by removing the eyes, and you can still see, or experimentally still see, the emergence of these functional cell assemblies. So this is something that puzzled us. And when we looked at the literature, especially in theoretical models of how these cell assemblies emerge, we saw a lot of studies that focus on this patterned external input that gives rise to these assemblies. But we know experimentally, at least from this example, that they really can come about in the absence of patterned input. So we asked what could drive the emergence of these self-connected assemblies in the absence of patterned input. And our hypothesis was that this is driven by internally generated network interactions. So what I will describe to you now is some work that we did that was primarily led by my postdoc, Lisandro Montangi, and my PhD student, Christoph Miel, that was published recently, that tried to answer this question. And when we started to look at recurrent networks and ask how could the emergence of these assemblies come about, we looked at sort of popular models of synaptic plasticity. And so one of these models of synaptic plasticity is spike timing dependent plasticity. So this is effectively the spike-based implementation of this popular Hebbian activity dependent plasticity that I described at the beginning. And so, I mean, I probably don't need to explain this, but according to this pairwise STDP rule, a synaptic connection between a presynaptic neuron J and a postsynaptic neuron I will undergo potentiation, or LTP, upon persistent presentation of a pre followed by a postsynaptic spike, and vice versa. If the postsynaptic spike precedes the presynaptic spike, then the synaptic connection will undergo LTD. So this was originally measured by B and Pu in '98 and has really been the subject of a large study of theoretical work that looks at what is the role of this particular plasticity rule in shaping non-random organization in these recurrent networks. However, if we think about how this pairwise STDP rule acts, if we have this one neuron A that persistently drives the firing of neuron B, because of this acausal or asymmetric structure of the pairwise STDP rule, it's not hard to imagine that this rule will always actually prefer unidirectional connections between two neurons. So you will always only potentiate the connection from A to B, but not the connection from B to A. So this rule is not able to generate these bidirectional connections, and these bidirectional connections are important for the generation of these cell-connected assemblies where you have many, many neurons that are connected to each other. And indeed, us and others have shown that this rule, indeed, when you put it in recurrent networks, cannot on its own generate these self-connected assemblies. But more recently, and this includes some work that I have done together with Jean-Pascal Pfister, is to consider that perhaps pairs of spikes are not the main building blocks for synaptic plasticity, but maybe the next contribution coming from triplets of spikes is important for driving plasticity. So this type of rule is called a triplet spike timing dependent plasticity rule because it relies on evoking potentiation or depression depending on the timing of triplets of spikes. So potentiation is induced by having two postsynaptic and one presynaptic spike. And so basically this third spike, which is effectively the second spike in the postsynaptic neuron, What it does is it modulates the amount of classical LTP that you will get from this classical pairwise STDP rule. And similarly, you can have a third spike, which effectively is the second presynaptic spike, which will modulate the amount of LTD or depression that you would get in classical pairwise STDP, the STDP rule. So this rule was proposed by Jean Pascal, and it was shown to capture a larger amount of experimental data than the pair-based rule. And especially these experiments where they induce plasticity by pairing pre-post, pre-post, pre-post spikes multiple times, but where they vary the frequency with which these pre-post spikes are presented. So you can have a scenario where you do pre-post, you wait for a long time, you do pre-post again, or you can have a scenario where you do pre-post, pre-post, pre-post at a very high frequency. And so these kinds of experiments can be much, much better described by this triplet rule. And so based on some preliminary work that we had done at the time, we postulated that this type of plasticity rule that relies on these triplet interactions might be capable of driving the emergence of these self-connected assemblies in these recurrent networks. And so this is what we started to do in this work. We consider the recurrent network of excited tree neurons, indicated here by the triangle. So these could be, for instance, the pyramidal neurons in the cortex. The connectivity in this case is all-to-all, but this is not that important. It could also be sparse and random. And these, we describe the activity of these neurons with inhomogeneous Poisson processes. So as we know, this is an appropriate representation if we study even, say, leaky integratin fire neurons that fire synchronously. But what is important for us is we use this framework so that we can calculate a lot of the contributions that emerge through the plasticity rule for the plasticity of the synaptic connections. So we use these Poisson neurons for convenience. And we drove this network with external input, but I should point out that this external input was completely unstructured. And so what I will show you in a moment is how these synaptic connections self-organize in a way that they build or organize into a non-random structure purely from internally driven correlations. We also had inhibition in the network, and this inhibition was not plastic, and it was just there, was always scaled to excitation just to prevent activity from blowing up effectively. So this, I should point out that mathematically we can treat this network using the framework of Hawkes processes, which are mutually exciting point processes. So what we can do is we can write the average rate of change in the synaptic strength between presynaptic neuron J and postsynaptic neuron I under the STDP plasticity rule. And we can do this by effectively writing the change in the weights as the product of the time-dependent cumulants of different orders. So these are these C's and K's that I will explain in detail in a moment, and the STDP functions. So the STDP functions here are denoted with the L functions. So L2 corresponds to the pair STDP rule, and L3 corresponds to the triplet STDP rule. And I have written here the equation in the most general form, where we explicitly write the contribution of all cumulants of second and third order as they combine with these two plasticity rules that use either pairs or triplets of spikes to induce potentiation or depression. So let's look individually at each of the terms that contribute to plasticity in this framework. We have two types of second order cumulants. We have first the pairwise cross correlations. So this is effectively the probability of joint spiking of the pre and the postsynaptic neuron as a function of the timing delay between the two neurons. And so as you see, the plasticity here, the contribution of this particular pairwise cross correlation to the plasticity comes both through the pair and through the triplet STDP rule. So it is integrated together with the two plasticity rules. But we also have another second order cumulant, and that is the pairwise autocovariance, the CII term, that only contributes terms in plasticity when it is combined with the triplet rule. So it is not present in the term with just the pair STDP rule. So this is a novel term that we bring completely from scratch in our analysis. And another term that we have, because we consider this triplet spike time independent plasticity rule because of the contribution from triplets of spikes, is this third order cumulant that I have denoted here in purple with Kij. And unsurprisingly, this term only appears in the second and third line of the equation here because it only contributes to plasticity as it is combined with the triplet STDP rule. So this is the equation for the weight dynamics in the most general form. I should just point out that due to other studies by Jean-Pascal, they actually showed that a more hybrid version of STDP, so one that only uses triplets of spikes for potentiation and pairs of spikes for depression, is just as good as capturing the weight dynamics in various experimental studies as the full triplet model that also uses triplets of spikes for depression. And so from now on, for simplicity, I'm going to be using this so-called minimal triplet model where, as I said before, triplets of spikes are only used for potentiation. So this allows us to slightly simplify the expression that we have for the overall weight dynamics by effectively eliminating this last line, which is the contribution of these triplets to long-term depression. So what can we do with this equation? So notice here, I have grouped the second and the terms in the second and first line according to the rules. So the first line is the contribution of the covariances to the pair rule, and the second line is the contribution of these cumulants to the triplet rule. But we can actually rewrite it so that we group it not according to the contribution to the rules, but according to the contribution of these different cumulants. So now each of the three lines of this equation denotes the contributions of either independent spikes, so completely independent of joint firing statistics such as correlations in the first line. The second line denotes the contributions of all these second order cumulants, so the cross correlations and the autocorrelations. And the last line gives us the contribution of the novel terms that are purely used by the triplet rule. So these are the autocorrelations and the third order cumulant. Okay, and I should point out that we make a simplifying assumption where we set this first term to be equal to 0 because we're only interested in how these internally generated correlations, namely the pairwise or these triplet correlations in the network, so completely intrinsically generated by the dynamics of the network, drive plasticity. So we are not interested in a regime where changes in firing rates dominate plasticity. So that's why we set this first term to be equal to 0. Okay, so what we can do now is we can actually rewrite this covariance term in this equation. And here I've only illustrated the pairwise covariance terms, so only the first expression in this equation. We can rewrite these covariance terms in terms of what we call structural motifs. So by doing this, we can actually break down these second and also third order cumulants into expressions that include the contribution of every spike that's propagated in the network through existing synaptic connections. So taking into account the full recurrent connectivity in the network. So we can take this cross covariance term, for example. So this is the expression indicated here with the asterisk. And we can write it by summing over all the possible paths from every possible source neuron K. So here we sum over the neurons K, and then we sum over all the possible paths that a spike takes from the source neuron K to the pre and post synaptic neurons I and J. And this is perhaps better illustrated by looking at this schematic. So we can write this Cij, this pairwise cross covariance shown here in this line of the equation, as the sum over all the spikes that go from the source neuron K and go either to the postsynaptic neuron I or the presynaptic neuron J via these two different paths that we call the alpha path and the beta path. And so I should point out that this framework was originally proposed by Ravi Tenenbaum and Burak for only the pair-based rule. And what we have done here is we have extended it to include terms also for the triplet STDP rule. So we have this now expansion, and we can do this also for this term and also for this other term. So for all the contributing cumulants of different orders. And what is interesting here is that we get these by rewriting the change in the synaptic weights in this way, we get an expression that depends on these so-called motif coefficients, which are functions, as you could see here, of the spike timing dependent plasticity rule, of the length of the alpha and the beta paths, and also of another variable that I haven't mentioned so far, but that is also important and is indicated here by this variable E, and that is the evoked postsynaptic current, or the EPSC. So it's the effect that a given presynaptic spike has on the sort of probability of spiking of the postsynaptic neuron. So we can go from this kind of equation into an equation where each of these terms can be rewritten by using these structural motifs together with the motif coefficients that tell us how strongly each of these structural motifs contributes to the weight dynamics. So let me give you some examples. So what I show you here are just some structural motifs under the pair-based rule that emerge when you decompose this pairwise cross covariance into these structural motifs. And so these are not new. These were previously proposed by Ravi Tenenbaum and Burg, but I just want to give you the intuition. So one interesting motif is this so-called 1-1 motif, where basically the alpha and the beta path have length 1. So here, always J is presynaptic and I is the postsynaptic neuron. And the reason that this is a 1-1 motif is because we have a source neuron K, indicated here in cyan, that projects with a single connection to the presynaptic neuron, and hence beta is 1, or to the postsynaptic neuron, and hence alpha is equal to 1. But we can also have other motifs where, for example, the source neuron is exactly the presynaptic neuron. So that's why here the length of the beta path is 0, because there's basically no connection onto itself. But there's a single connection from the presynaptic neuron to the postsynaptic neuron. That's why alpha is equal to 1. Or we can have a different motif where now the postsynaptic neuron is the source neuron. So here the alpha path is 0, but the beta path is 1, because we have this path that can influence this connection from the postsynaptic to the presynaptic neuron. So I hope this is at least intuitive. Yes. 5. Okay, good. So I hope this is intuitive, at least for how to rewrite these contributions for the pairwise cross covariances. But let's actually look at what are some of the novel structural motifs that emerge under the triplet STDP rule. So what we have here is a way to rewrite the new term that emerges under the triplet rule. So here we have the triplet rule as L3. So we can rewrite this pairwise autocovariance again as a sum of these structural motifs. And what is interesting here is, in addition to the standard alpha path, where we influence the neuron by looking at the influence of spikes from all other source neurons in the network, we have basically an identical path from the same neuron, source neuron, to the same postsynaptic neuron, but now via a delay. So this is now the contribution. So by introducing these triplets of spikes and by allowing to kind of take into account more than just a single postsynaptic spike in the induction of plasticity, we can now include paths that sort of structurally take the same path through the network. They go through the same neurons, through the same connections, but with a given delay. And so this is a new novel term that, as we show in a moment, plays an important role in determining what happens in the network. So what I will not show you now, but we go into detail about this in the paper, is how to also decompose these third-order cumulants in the same way using these structural motifs. So let me just give you some examples of these motifs when we introduce the gamma path. So here again, we have a presynaptic neuron J and a postsynaptic neuron I, and we have a source neuron K. So as before, we can go into the presynaptic neuron via a single path. So that's why beta is equal to 1. But we can now go to the postsynaptic neuron sort of via two connections, a direct one, which is the alpha equal to 1, and a delayed one, which is the gamma equal to 1. We can also look at two other interesting motifs that come in this framework that for sure do not affect the pairwise covariance, and therefore we know do not play a role in the induction of plasticity under the pair-based rule. And these are very interesting because they completely do not affect this presynaptic neuron. So these for sure cannot exist in the case when we study the pairwise STDP rule. So here, either the source neuron is either the postsynaptic neuron, so we kind of have this interesting loop that goes back to some source neuron in the network and comes back to the postsynaptic neuron again. Or we have this other scenario where we have a source neuron that again projects with this delay into the postsynaptic neuron. this particular motif that I showed you here, where we have the postsynaptic neuron to also be the source neuron that has these interesting loops, we call them loop motifs, is actually very important for the formation of these assemblies. So I'm just gonna flash this very briefly to show you that what we have achieved here is we have found a way to decompose completely the equation for the induction of synaptic plasticity, where we had the contribution of all these covariances, so the second order cumulants and the third order cumulant, as effectively a linear combination of these structural motifs. And so these are just the examples. You can study them. We can write them exhaustively for the case that we consider. The really interesting ones, however, are the ones in the second and the third row, because these are completely novel structural motifs that emerge under the triplet STDP rule. And especially interesting are the ones that I have circled here in gray, because these are the ones that we call loop motifs that I will show you in a moment give rise to the emergence of these assemblies. Okay, so how are we going to study the emergence of these assemblies? We're going to look at the triplet rule again. So we have the potentiation part, which is evoked by triplets of spikes, and we have the depression part, where we're going to modulate the time scale of, or effectively the influence of depression by broadening this influence through the time scale of depression. And this has been motivated biologically in various experiments where, for example, the application of different types of neuromodulators can indeed deepen this influence of this depression. And so we can look at the evolution of these different types of motifs that we have in the network. And so here we have the motif coefficients as a function of how deep this depression is in our plasticity rule. So classically, people think that this common input motif, this 1-1 motif shown here in the green, is the one that actually drives the emergence of assemblies. But what we find in our work is that although this motif is important, you see here, it's not the dominant motif. What ends up driving the formation of assemblies, self-connected assemblies in our work, as we show here at the bottom, as we vary this depression strength of the plasticity rule, is the relative interplay of two types of motifs. We call them feed-forward versus reciprocal motifs. So the feed-forward motifs are ones where the alpha path, so this is the one that goes into the postsynaptic neuron, is longer than the beta path. So here with this blue ellipse, I'm trying to indicate all the feed-forward motifs in our framework. But we also have these reciprocal motifs. And actually what we see at the bottom is as we vary this depression strength of the plasticity rule, we see that these reciprocal motifs, this is basically a zoom in on what really happens in this plot. We see that these reciprocal motifs cross from being negative to being positive right around the values of this depression parameter eta minus, where we see the emergence of these assemblies. And so when these reciprocal motifs are negative, assemblies cannot form. So these are the cases for very weak depression in the network. But as these reciprocal motifs turn positive, then they can effectively support these bidirectional connections that are important for the emergence of these assemblies together with the feed-forward motifs. Okay, so I just want to summarize the emergence of these assemblies using a graph theoretic measure known as the clustering coefficient, which tells us here exactly how these, or quantifies how strong or how tight these assemblies are as a function of this modulation parameter. And I'm going to end by really stressing the importance of the novel terms that we have derived under the triplet STDP rule that are important for the emergence of these assemblies in contrast to the pair rule. So if we only consider these motifs shown here in the top row that also take part in plasticity under the pair rule, we basically see very weaker or not as strong clustering shown here with the blue curve when we apply it in our plasticity framework. What we need to do is include some of these terms that emerge uniquely under the triplet rule, and especially these so-called loop motifs. So here I include the terms that participate in plasticity with the pair rule, and only these two novel terms that we call loop motifs. And if we compare the performance with just these motifs, we see much weaker clustering in these self-connected assemblies. But if in addition we add these loop motifs, we see much better clustering in these self-connected assemblies, even better than actually when we include all the terms in the network. Okay, and one can actually visualize what this connectivity looks like as a function, if one looks at these connectivity matrices as a function of this modulation parameter and plasticity. Okay, so I'd just like to summarize with what I have shown you. I sort of divided my talk in two parts. In the first part, I explained to you how we managed to derive a framework that gives rise to plasticity in recurrent networks that includes second and third order correlations. So we utilize not just the pair STDP rule, but the triplet STDP rule that utilizes the contributions from these third order correlations. We showed that we can rewrite these contributions from the cumulants through these structural motifs, and that we showed which particular structural motifs that are novel under the triplet STDP rule are important for the generation of these self-connected assemblies. And at the very end, I just want to thank the people who did this work. This was my postdoc, Lisandro Montangi, who did an incredible work of plowing through these calculations together with my PhD student, Christoph. And I'm happy to take questions in the remaining 2 minutes. Thank you.
Thank you. Very interesting presentation. So I will go and turn the first comment into a question. This is from Mark van Rossum. This is Song and Abbott in 01 show that recurrent connections can arise with pairwise STDP as long as the time constants for LTP and LTP are different. So the question in more general would be, can you get assemblies with pairwise rules?
So the question was that Song and Abbott showed that you can get bidirectional connections under the pair rule if what is the case if you have?
So the
time constants are different.
and LTD are different, right? So it's a comment, but I'll turn it into a question. So yeah.
No. So as we show here, if you only use the pair rule, so let's look at this here. If we only use the pair rule, and if we look at the contribution from only these pairwise cross covariances, we cannot actually get these self-connected assemblies. Here, indeed, we are increasing the dominance of depression. That's what we're doing by this depression parameter eta minus, and we cannot get as good assemblies that look as tight as what we can get with the triplet rule. I mean, so Ravid, Tenenbaum, and Burak argued that you can get assemblies with the pair rule, but they had to assume two things. They had to assume a symmetric STDP rule, so they had to basically eliminate this causal or this asymmetric property of the STDP rule, and they had to introduce synaptic delays. So they had to do these two tricks in order to get assemblies with the pair rule. But as we show here, you cannot get and in that case, I should point out that when you do get these assemblies with the pair rule, you get them by amplifying this so-called this common motif, so this 1-1 motif. The way we get assemblies here is very different. Here we get assemblies by promoting what I call these feed-forward motifs over the reciprocal motifs. And so this is interesting because it's a different way to get assemblies. And it's also interesting because the assemblies look different. If you just promote this common input motif, you get perfect assemblies where one assembly is self-connected within each other and completely disconnected from the other assemblies. But by doing this in our way with these feed-forward and reciprocal motifs, you can get assemblies that are perhaps more biologically realistic because they're not decoupled from each other.
Right, thank you. To keep on time, we'll go to the next talk. Okay. I think there's a number of more questions. Some of them are just clarification, which you could probably answer in the chat box. But we'll move on to Robert Rosenbaum, who's in the Applied Mathematics Department at the University of Notre Dame. And go ahead, Robert.
Thank you, and thanks for inviting me here. Or here, I didn't go anywhere, I guess, but okay, so today I'm talking a little bit about universal properties of strongly coupled networks. This is the term that's used in the literature for it, but really what I mean is not so weakly coupled networks, and I'll clarify that in a little bit. They're not actually all that strongly connected. So the idea is that cortical neurons receive hundreds or maybe thousands of synaptic inputs from other neurons, either in other layers or other cortical areas or subcortical areas or from within their own layer. But postsynaptic potentials, each postsynaptic potential pushes the membrane potential, around 1/30th or 1/50th or so of the distance from reset to threshold. So in this sense, the networks are not all that weakly coupled, right? They receive far more inputs than are needed in terms of the scale of the distance from rest to threshold. So to formalize that mathematically, just consider a large network. For now, let's not even specify how the membrane dynamics or firing rate dynamics evolve. Let's just consider a large number of excitatory and inhibitory neurons that are recurrently connected and receiving input from some external layer. We use INs to denote the size of the networks. And then the synaptic input to one neuron, this would be neuron J in population A in this equation. It's written as a sum of synaptic weights across other neurons. And then for a spiking network, you might have a postsynaptic current or postsynaptic conductance waveform summed over spike times. For a rate model, you would just sum the rates, right? So the idea is there's this inner sum, this sum over n. So in general, neurons can receive a lot of inputs because n are large. The networks themselves are large. More specifically, following from that equation, one would come to expect that the synaptic input to a single neuron is order j times k, where j is a typical synaptic weight, k is a typical n degree, or if we want to consider randomly connected networks, it'd be j times the probability of connection times the number of neurons. Of course, if there's multiple populations, there can be multiple probabilities of connections or n degrees from different populations, but this is the general idea. But we don't want I to explode, right? We want to consider large N, large networks, and we don't want the synaptic input to any individual neuron to become very large. So classical methods for avoiding large synaptic inputs are to consider sparse coupling. This would be taking connection probabilities that are 1 over n, or n degrees that are order 1 as n gets large. Or weak coupling, this would be scaling synaptic weights by 1 over n and keeping connection probabilities order 1. But we argued from the beginning that maybe cortical circuits aren't all that weakly or sparsely connected. That was my first slide, right? So the question is, can we maintain sort of reasonable magnitudes of synaptic inputs in large networks without assuming sparse or weak coupling? So why don't we just let this product of J and K be large and see where it takes us? So now let's just assume that neurons receive a lot of inputs that are not all that weak. So let's clarify here that we're not assuming that individual synaptic weights are large. We're not even assuming that they have to be order 1 as n grows, or you can assume here that n is growing or k is growing. We're going to grow these proportionally in the models we consider here. We're only assuming that they're larger than 1 over k, or larger than 1 over n, however you want to look at it. In other words, the network is not so weakly coupled, right? So we don't scale synaptic weights like 1 over k or 1 over n. And this is sort of borne out to some extent by some experiments in culture by Alex Reyes, who found that if you grow cultures of cortical neurons and increase the density of neurons, effectively increasing the number of neurons in the culture, the number of connections increases proportionally to the density. So that means the probability of connection is approximately order 1, but the strength of connections, so the postsynaptic potential amplitudes that they see, actually decay. So they get smaller as the network becomes denser and more neurons receive more inputs. But they don't become small like 1 over k. So the best fit power law to this is 1 over k to the power of 0.6. So I'll be more specific about that in a little bit. So balanced networks take a scaling where J does not scale like 1 over k, where jk is large. And balanced networks, if you're unfamiliar with them, synaptic weights scale like 1 over the square root of the n degree, 1 over square root k, which means that this jk bar parameter I've defined, which kind of quantifies how strongly the network is coupled in a sort of mean field sense, is order square root k. Or if we take n to infinity instead of k to infinity and fix connection probabilities, then the n degree is order n, k is order n, and the product of j and k, which we're sort of using to quantify the coupling strength, is order square root n, and we can take n to infinity. We're going to kind of consider these two limits to be equivalent because we're taking connection probabilities to be order 1. So as I mentioned before, I accidentally swapped the order of these two slides at the last minute without realizing I would scramble some things. But Alex Reyes showed that this is approximately true in cortical cultures. He found that synaptic weights scale like approximately 1 over k to the 0.59, whereas 1 over square root k would be 0.5. So the best fit decay of synaptic weights is very close to what you see in balanced networks, a little bit faster, but not 1 over k, right? The slope would be -1 if it were 1 over k, which would be a weakly coupled network. Finally, balanced network models, when you do scale synaptic weights like this, reproduce a lot of features of real cortical circuits. So you get asynchronous irregular spiking, you get spike train correlations that have a mean that's very close to 0 and are distributed in a sort of symmetric way around 0. This was first shown by Alfonso Renart and Jaime de la Rocha and their colleagues in 2010. You get EI tracking in the sense that excitatory and inhibitory currents, inputs to a neuron sort of track each other. They're highly correlated. And E and I are anti-correlated if you take their sign into account, the polarity, which is what we see in a lot of recordings. We also showed that in these balanced network models, you get distance dependence of correlations. So the decay of these weak correlations with respect to the distance at which neurons are from each other is reproduced in the models like you see in experiments. Okay. So just to qualify this though, in reality, this JK bar might not be all that huge. Ahmadian and Miller recently argued in Archive that it's not all that large. Of course, it probably depends on species. It might be different in mouse and primate, might depend on cortical layer, cortical area. Depending on how it's measured, this could depend on behavioral state or sensory state, something like that. But I just want to argue that the large JK limits, these not so weakly coupled networks or strongly coupled networks, if you want to call them that, provide a decent first approximation to cortical circuit responses. And today I'm going to argue that they provide a mathematically rich approximation, which is something that this ICMNS community presumably likes, and that it's mostly model independent. That is, we can look at the network architecture and understand about approximately what the network is doing without having to specify exactly how we're modeling individual neurons. So that's the argument I'm going to make today. So the first part of my talk, I'm sort of on a soapbox. Why should we be modeling circuits like this? And the last part of my talk, I'll talk about some results. Okay, so let's transition to the mean field. Let's say we have one of these not so weakly coupled networks. So the recurrent connectivity, the recurrent input to a neuron, so I here is the average input to each population in the network, is going to be order JK bar, like I said, and JK bar is going to be large. Similarly with the feed forward input, which I'm denoting with a subscript X here, this is the external input to the network. We can pull that JK bar out to kind of keep track of the fact that it's that it becomes large and everything else is order 1, all the other parameters. And it's long been known that in this limit, the strong coupling limit, that rates are approximately a linear function of external input. So they can be written as negative W inverse X here. The idea is that if JK becomes large and you want the inputs to not become large, you need a cancellation inside that square bracket right there in the first equation. I think I can actually point to this. You should be able to see my pointer here. In fact, you can prove this if you want. It's actually not that difficult to prove at all. If you want I to be order 1, then you need the limit as JK bar goes to infinity of R to be this function. So that's a very nice, simple framework. It applies to rate network models. It applies to spiking network models. It really all depends on just the linear algebra of this equation. It's not even a very complicated concept to prove. Also, it generalizes to any number of populations, so we don't have to consider just one E and just one I population, and to spatially extended networks. Here are some examples from some papers that I've worked on with my students and an old paper with my postdoc advisor where we tested this approximation in various networks. So here on the left is a network of exponential integrating fire neurons with 4 populations, 2 excitatory, 2 inhibitory. In the middle is a network of adaptive EIF neurons with 2 populations, 1 excitatory, 1 inhibitory. And all of these plots, the solid curves are from spiking network simulations, and the dashed curves are from that equation I showed on the previous slide, or the equation that's right here in the center of this slide. And then the spatially extended example, with LAS, we pushed the number of neurons up to 2 million. So we simulated 2 million LAF neurons and found that this, in the spatially extended case, even this approximation for R is pretty accurate, and the large N limit. So in all of these cases, we're looking at how accurate it becomes as N grows large. Okay, so there's some problems with this though. So this equation has been known in various forms for a while, but there are some problems with it. The first is that in this equation, X here is the external input to the network. So it's like a stimulus. W is the mean field connectivity matrix. So it's the matrix of average connection strengths, you can think of it, between neurons in different populations, right? And R is the vector of population average firing rates. So this tells us that the population average firing rates are a linear function of the stimulus, at least under this approximation, but we know the cortex can perform nonlinear computations, obviously, it needs to. Another problem is that under this equation, parameters need to be chosen to produce non-negative rates. Now that's relatively easy when you have one E and one I population. When you just have two populations, then you need to satisfy two inequalities that are relatively well known. They were in the original Van Riesvik and Sompolinski paper, and it's easy to choose parameters such that that's true. However, every additional population you add onto your network adds another inequality to satisfy, and the proportion of parameter space that satisfies all of those inequalities is then going to decrease exponentially with the number of populations. So it becomes more and more difficult to satisfy this inequality as you include more and more populations in your network. And my student, Vicky Zhu, was able to prove actually even more sort of damning is that for any connectivity matrix W satisfying Dale's law, there's a positive stimulus, a positive X, for which this expression, negative W inverse X, has negative entries. Remember, negative entries are things that break balance. That means any connectivity matrix that satisfies Dale's law admits a stimulus that breaks balance. So while simple and somewhat model independent, this equation has these weaknesses that means maybe it doesn't account for everything we sort of need it to. Okay, so to demonstrate these notions, here we simulated a network of 30,000 EIF neurons, 2 excitatory populations, 1 inhibitory population. They're receiving external input from 2 external populations. There's connectivity between all the recurrent populations and from the external to all the recurrent. And for the first 500 milliseconds of the simulation, you can see the raster plots of a subsample of the neurons on the left, voltage trace of 1 neuron. In this case, we put the network, we chose an external stimulus so that the balance state exists. And you can see everything sort of works out nicely. You get a balance between excitation and inhibition and the inputs to all 3 of these populations. But then we transitioned to a stimulus for which this balance state does not exist. So this equation that I wrote before, R equals negative W inverse X, predicts that some population, one population, E1 in particular, would have a negative firing rate. So it can't possibly be valid. And what we see in this case is maybe not so surprising when you think about it. That population turns off. It just doesn't spike at all. You can see that in the raster plot on the left. It receives excess inhibition, so it's breaking balance here. But the other 2 populations then form a balanced subnetwork. You can basically forget about population E1, and now we just have sort of a standard balanced network with populations E2 and I. So this is sort of an illustrative example of what I refer to as the semi-balanced state. So the semi-balanced state occurs when balance is broken. So when you have this balance equation predicts 1 negative rate, which gives rise to excess inhibition to some populations, and the remaining populations form a balanced subnetwork. Okay, and it's very simple when there's just a few populations, but it's actually more difficult to work out what exactly is going to have when there are more populations. So my student, Vicky Zhu, again, was able to derive an equation for firing rates that generalizes this W inverse X equation that I was talking about before for balanced networks. She was able to generalize this to the semi-balanced state with a couple more assumptions. So here we have to assume an FI curve, but we only place relatively weak assumptions on the FI curve. And we find that in the semi-balanced state, we get firing rates are satisfied by this equation here in the gray box, where the brackets plus is a rectified linear threshold linear function. So you can kind of see if you think about it for a minute, if R is positive, if WRX plus R is positive, then this is going to reduce to WR plus X equals 0, which gives back the same balance equation we had before. So this generalizes that. So when all the firing rates are positive, this gives the same state. But when some firing rates are 0, it gives a different state. This could admit multiple solutions as well, unlike the standard balance equation. Well, here's a more direct comparison. And the assumptions, the main difference in the assumptions between the two theorems is that for balance, on the bottom here, we assume that the input to all neurons remain bounded. For semi-balance, we assume that none of the firing rates approach the maximum of the FI curve. So that could be infinite if you have an FI curve that goes off to infinity, or it could be finite if you have a saturating FI curve. So if you assume that you have a strongly coupled network, and you assume that none of the rates blow up and saturate, then the rates have to satisfy this equation and the strong coupling limit. Whereas for balance, we assume that the inputs remain finite. The difference is in the semi-balanced state, some of the inputs can diverge to negative infinity, which just turns off those populations. One thing that's nice about this equation is it gives you a nonlinear mapping from stimulus to rates. So now our networks can implement nonlinear functions, which was a weakness of the balanced state that they couldn't. Another interesting thing about this equation is that it maps directly to fixed point rates in RNNs with rectified linear activation functions, which are equivalent to these threshold linear networks that Karina Courteau and her colleagues study. So you can just adjust the connectivity matrix by adding one term, and you get back exactly what Karina Courteau studies. And she's shown that these networks can exhibit some very rich and interesting properties. So there's a lot to study here. And now we've connected that to any sort of strongly coupled network. It doesn't have to have a threshold linear activation function, and it can be a spiking network, whatever you want. And it provides a decent approximation for networks with large n. So again, we're always looking in this sort of large n limit, and biologically realistic coupling strength. So we simulated networks of adaptive EIF neurons, Izakevich model neurons, adaptive EIF with conductance-based synapses, for which we have to make some adjustments to the equation, but it still works out. And these based rate networks and a stabilized, super linear stabilized state studied by Ken Miller and colleagues. Okay, so it gives a decent approximation, and it's an interesting equation. So far though, I've only talked about balance and now semi-balance at the level of population averages, right? This is to say that so far I've talked about if you average the input to neurons in a population, the average excitation to the population is going to approximately cancel with the average inhibition. But now let's transition to talking about what's called detailed balance, which is to say that every single neuron in the network receives a approximately balanced input. Okay, so to demonstrate this idea of detailed balance, I ran a simulation of a network with just one E and one I population now. And for the first, I think it was 40 seconds of this simulation, the left plot of this time series here, this was just a randomly initialized network, nothing special about it. And you can see here that excitation and inhibition, so these are each red line here, each red curve is excitation to one neuron in the network. I think these are strictly excitatory neurons, but it doesn't really matter. Each blue curve is inhibition, and the black curves are the total input. And you can see that the network is not really balanced, right? The spread of the total inputs is similar to the spread of the excitation and the spread of the inhibition. So it was shown by Tim Vogels and colleagues in 2011 that inhibitory synaptic plasticity, homeostatic inhibitory synaptic plasticity, can achieve detailed balance. So after 40 seconds of the simulation, we turned on inhibitory synaptic plasticity, and indeed, we see that the network achieves detailed balance. So all the neurons receive balanced inputs, right? And you also see that the rates sort of collapse to this target rate that's defined by this STDP rule. So this is a classic picture, well known by now. You can also see it quantified here. This is a histogram in log space, so with a logarithmic vertical axis of the total current. That's the black time series in the right plot. And before synaptic plasticity is turned on, you see this very broad distribution of total inputs. After synaptic plasticity is turned on, you see this very narrow distribution of total inputs, meaning that excitation and inhibition are precisely canceling on a neuron by neuron level. Okay, but this is for one input. This is for all the neurons receiving the same synaptic input, external input, sorry, one stimulus. So the whole network's just receiving a time constant stimulus. If we now start changing the stimulus, so what we did here, you can see in this network schematic to the left, what we did is we basically, every second, I think, or 2 seconds maybe, we randomly sampled a stimulus from, so the stimulus space now is n-dimensional, the dimension of the number of neurons in the network. Here there's 5,000 neurons in this network. We sampled a new point in this 5,000 dimensional space, but we restricted ourselves to a 2-dimensional plane. So we're basically sampling points on a square in this 5,000 dimensional space. And when we did that, what we see is we no longer exactly get detailed balance. So here, just looking at the time series first, we see these deviations in the total input, but only more or less only in the negative direction, right? So you see these negative deviations of the total input. And if you look at the distribution, that's very clearly borne out here in this heavy negative tail in this purple histogram here. So what that tells us is that this network is, and we also see firing rates that hit 0 every now and then. And this tells us that this network is in a detailed semi-balanced state, in the sense that you never receive excess excitation, but some neurons receive excess inhibition in response to some stimuli, and those neurons are silenced by those stimuli. And the stimuli change, and it's another subset of neurons that are silenced and receive excess inhibition. This is the defining feature of the semi-balanced state. So that is to say, networks with homeostatic inhibitory synaptic plasticity, if they receive input from, if they receive one stimulus, and this is actually the scenario in which they're very often studied, then you get detailed balance. And that's been known. But actually, if you start changing the stimulus, and this is a relatively low dimensional stimulus space we're sampling, just a 2-dimensional stimulus space, the network's unable to cancel, to learn to balance all of the inputs in this 2-dimensional stimulus space, but it can still semi-balance these inputs. So the network naturally finds this detailed semi-balanced state in which every neuron in the network is in a semi-balanced state. Okay, so let's explore this state a little bit. So this is these are simulations from the same network I showed on the previous slide. And this detailed semi-balanced state, we have plasticity turned on, inhibitory plasticity turned on, except now we started sort of exploring as we trace out, remember this is a 2-dimensional stimulus space, as we trace out this 2-dimensional stimulus space, which now we're tracing it out on a lattice instead of randomly sampling the points. What is the representation of firing rates of these stimulus? And we can explore that by looking at the neural manifold. So as we explore this 2-dimensional stimulus space, how do the firing rates change with respect to one another? And this is just 4 of the astronomical number of neural manifolds there are in a network of 5,000 neurons. And we see that they are nonlinear and they're highly intricate, right? So you can get all sort of, it's kind of fun actually to play with these simulations because you can just plot all these neural manifolds and see all these sort of exotic looking shapes. So you get these complex, intricate nonlinear neural manifolds in the semi-balanced state. To quantify how nonlinear they are, one approach is to apply PCA versus isomap. So PCA finds a linear manifold that captures the variability in the data, whereas isomap can find a nonlinear manifold. So the amount of variance explained by PCA versus isomap will be different if the representation is nonlinear. And indeed, we see that the PCA needs around 8 dimensions to capture all the variability, whereas isomap captures it in 2, which is what we would expect because there's a 2-dimensional stimulus. So the neural manifolds are necessarily explained by 2 dimensions. It's just a question of whether isomap finds that 2-dimensional representation, and it does. Okay. Finally, we wanted to test sort of the power and the potential utility of these nonlinear representations in the semi-balanced state. So we took the same two population network, one E population and one I population, and instead of giving it sort of random stimuli, we gave it stimuli that were projections of MNIST images. So the actual pixel representations of MNIST images, we just projected them onto the excitatory neurons in the spiking network. And what we found then is if we look at the firing rate representation, now we can take these excitatory neurons and look at their firing rates. They're going to have, there's one vector, one NE dimensional vector for each MNIST digit. And let's look at the representation of those MNIST digits in the excitatory neurons firing rate space. What we find is that in this firing rate space, I'll kind of go through this quickly because we're running a little low on time. In the firing rate space, we find that this representation is linearly separable. So we can train a linear readout of the firing rates that perfectly classifies the 10 different MNIST digits. But if we try the same linear classification in pixel space, it doesn't succeed perfectly. We get some discrimination, but there's some misclassifications. That is to say, this nonlinear representation helps with the classification of MNIST digits. And this isn't necessarily surprising. A random nonlinear representation, we have actually found, now that we're addressing reviewer comments, a random nonlinear representation does just as well. But this is just to demonstrate that the nonlinear representation implemented by these networks are actually doing something potentially useful. And to take that one step further, we trained another spiking network, or we implemented another spiking network that reads out the correct labels for these MNIST digits by training this linear classifier and then setting up a winner-take-all network. Don't really have time to go through it, but you can see here that the layer 2 raster plot indicates that the layer 2 is spiking in response to the correct digits, basically. The layer 2 population that spikes the most corresponds to the population with the correct label. Most of the time, it gets some of them wrong. Okay. So just want to make one conclusion. I'd argue that this is sort of somewhat preliminary work in the sense that I think it's a complete work, but there's a lot of directions to go from here. So the main conclusion I want to draw is that not so weakly coupled networks that are large N, and that have time various stimuli naturally operate in a semi-balanced state. And firing rates in this state are approximated by this equation. And this is a useful equation in the sense that it's, so the universal word in my talk title is to say that this is model independent. It doesn't matter what type of models you're using for your neurons. This should give you a relatively good approximation and not so weakly coupled networks. And I think there's a lot to explore here. So this is sort of just a first study of these ideas. And yeah, I'm happy to talk more about different directions to take this. My current graduate student, Vicky Zhu, is extending this to networks. Well, I guess these already have plasticity, but what I meant to write here is with learning. So looking at how these expressions can help us understand how to train spiking neural networks to actually learn useful tasks. And if you want information, more information on this, you can see our bioRxiv paper right here. And thank you for listening.
Thank you. Very interesting talk. So we'll have time for one question. And so what we're going to do is the questions also, there were a lot of questions from previous talks as well. They're all on Slack right now. So I hope that Juliana will have some time to answer them on Slack. So there's this is an ICMNS Slack team with different channels. There's a channel devoted to this particular session, and the questions are there right now. So Robert, how were the MNIST digits projected to the E neurons?
So we've tried both a random projection and a projection in which, we took a subset of the MNIST pixels. So the images are 20 by 20. So there's 400 pixels and 4,000 neurons. So we did one example where each pixel projects exactly to 10 neurons. And we did another example where we just multiply them by a random matrix. And we get similar results either way.
All right, thank you. So in the interest of time, we'll move to the next speaker. So the next speaker is Omri Barak, who's in the theoretical neuroscience group at Technion. And Tilo.
Hi, let me see. Okay.
All right, Omri, we can see you. Yeah, you just need to share screen. Yes.
Yeah, I'm trying to share the screen. Okay, can you see the screen now?
Yes.
Okay, good. Thanks a lot for the invitation. It's this strange concept of digital conferences. And so what I wanted to share with you today a bit is about structure and randomness and trained networks. And let's see how this all connects. Yeah, if there are questions, then just let me know. So first, I want to linger a bit about the title of my talk, this structure and randomness, and it sort of seems natural, but in a sense, one can ask why do we want this? Why should we care about structure and randomness? And it has sort of a normative flavor to it. Sounds a bit like order from chaos. You can even think of biblical connotations, understanding from confusion, or simplicity for complexity. All of this is, in a sense, what science aims to do, right? We observe complex phenomena and we try to make simple explanations of them that sort of capture the essence. In a sense, models are all about extracting structure from what will be an apparent randomness or something that is complex, and we want to try and understand it. And but the word simple, of course, is not well defined. So one could think of simple models as those that explain a component of the system. Now, this component doesn't have to be, it can be a physical component, but it can also be a functional component. So it can be a certain function the system has, or an actual anatomical component. And then the idea is usually that if we understand this component in isolation, and later we embed it back into the full system, functionality is retained. I mean, it can, of course, interact with other components, but this essence that we understood of this component in isolation, the simplicity of it, still helps us to understand what goes on when we put it back into the full system. But is that the case? So in neuroscience, at least, one common theme, that one common component is a single neuron. So when people look at single neurons, for instance, in V1, and have studied them for quite a while, even before 2004, then there were receptive fields and orientation selectivity. And when people try to understand this famous work by Olshausen and Field, how much do we, so if we know this simple part, if we know how the visual cortex neurons respond to all the stimuli that we gave them, and now we try to understand, let's say, natural vision with it, how much do we know? And their conclusion was that not much. In the auditory cortex, a very similar conclusion was reached when trying to look at the auditory receptive field, the spectrotemporal receptive fields, and then looking at more natural stimuli. And again, this didn't go so well. And this sort of goes on and on. So work that I was involved with by Mattia Rigotti and Stefano Fuzzi, using data from Early Mirrors Lab, then you could see that neurons respond in a selective manner to certain visual stimuli, but this selectivity is highly modulated by the context. So the neuron can respond if this is in the context of recognition task, but not in the context of recall task. So had you only observed the neuron during a recognition task, you might have reached the conclusion that you know what this neuron does. But now when you move to another context, suddenly the information gained in context A is no longer so valuable. This is also true in work that I did analyzing data from the Romo lab, where this tuning curves were done both during the stimulus and during a delay period. And you can see that while there is a correlation, about a third of the neurons sit sort of in the wrong quadrants. So a neuron that is positively tuned in one epoch of the task is then negatively tuned in another. What all of these examples show is that these parts or these components that are often used in neuroscience do not retain their properties when they are put together into the big system. And that sort of leads us to search for new simple parts. And what I want to talk about today is one type of approach that might lead to such new principles, to such new structures in the complexity. So here's a caricature of how one might use simple parts to construct a model. And it goes sort of like this. You observe a behavior, let's say a certain task, and then you that has a computation involved, or some sort of a, in a sense, you could say there's a problem that the animal solves. And then in order to understand how the animal solves this problem, you identify the relevant components. Let's say if it's identifying a visual image, then you might say, well, the neurons that observe that are sensitive to various orientations are definitely relevant components, right? So visual cortex neurons should be relevant to identify a car from a cat. And now, using some insight, you try to think how these components that you identified and characterized are connected together to support the functionality. This is a hypothesis. And now, let's say if you're in the business of high-dimensional dynamics, then you put this, you implement this in a neural network that has many neurons and has certain dynamics that you can then compare to neural data. And in recent years, there's another approach, which is the machine learning approach, which sort of throws away all of this identifying and insight and understanding how things work, and just says, well, let's take image recognition or memory, and let's just have a network and turn the crank, and out comes a network that performs a task, and let's compare that to neural data, with of course this small caveat that we skipped the whole insight and hypothesis phase, and therefore one might think that we have no clue what we ended up with. Luckily, in some cases, it is possible to reverse engineer this and sort of discover the underlying hypothesis that came about here. And so this could seem that a bit of a let's say highly indirect approach of doing science, and I want to slightly try to convince you that it's worthwhile. So why train networks? And basically two reasons. One, I think it fits the data better than the sort of standard models. And two, we can learn something from it. Ideally, these new forms of understanding, we might be able to learn them. So it fits the data better. Actually, I will devote very little to this today, but this is both of my work and others. This is an example from the Romo data. So here is the scatter plot I showed you before of consistency of tuning curves. So this is the tuning curve during a stimulus versus during the delay, and you see the scatter plot. The curves here try to sort of do this over time of many points compared to one another. And you can see that, let's say, a network that has the correct components wired together in the correct way will have, by definition, very consistent tuning, whereas a trained network will have a profile that is more similar to that observed in data. Similarly, this is from the Ganguly lab, where they trained networks to replicate the retina response, and that was closer to the actual physiology than relying on sort of more classical tuning curves. And this has been done in many areas by now in motor cortex, using timing, vision, auditory. And the gist of it, I would say, is that these type of networks naturally capture, let's say, the messiness of the biological recording. That when you start with sort of if you look at 1,000 neurons and then you try to make sense of them, then you might be biased to choosing sort of the clean ones and the ones that make sense individually. And then you build a model by connecting those nice neurons to one another, and you end up with a nice network. When you start the other way around with a random network that you train, then if it works, you get something that is both messy and therefore maybe like biology and also works. But this, you could just add noise. I mean, why go through all this trouble? So hopefully we can also learn something in the process. And so let's try to understand these networks. And so one analogy that I think Robert Young made, and David Cecilio and others, is that you can think of these as a model organism. So that's sort of just one thing to keep in mind, which I think is nice, that in biology we have many models, right? So a mouse is sometimes a model, a human is sometimes a model. I mean, many things are a model. And whenever you make a model, either biological or in silicon, you sacrifice some realism and you gain tools. And my claim is that obviously you have more tools when the entire network is at your disposal in the computer. And my claim is that you don't sacrifice all of reality in the process. And when you study this, there are two sort of fundamentally different or conceptually different things that one can study. One is the final network. So you have a task, you train a network, and then you observe and analyze the final outcome of this. The other is the learning process itself that's also accessible to you. And for instance, you could be surprised by the solutions a network finds to a task. Maybe this is something you did not think of. Or even if you're not surprised by the result, you might gain some insight on the process of learning itself. And there's a nice citation, a quote from Richard Sutton, where he said that if we build in our discoveries, it makes it harder to see the discovering process itself. Okay, so if we study the process of learning, then that can also give us some insights. And I'm going to partially talk about both things now. Okay, so I want to briefly describe two projects that touch upon these things. And the first is by Tia, a recent graduate from the lab. He finished his PhD this year. And this was virtually published in ICLR. And the task is the following. This is a navigation task inspired by the water maze. So there's an agent that is in a square arena, and there's a certain goal. The agent starts in some random initial position, and it is controlled by a recurrent network that is initialized in a random hidden state. And there's a context which tells the agent which of two different targets are active in the current trial. The agent only receives proximal input, so you can think of it as somatosensory if you're in the dark. And then it has to move about and locate the target. And obviously it has to bump into the walls to know where it is. And our focus here was on what types of internal representations are necessary to solve this task and how they may come about. So what we did to that is we did a two-stage learning. So first, we just let the agent run around in the maze, in the arena, without a task. So there was no goal. And this was sort of akin to evolution or development. There was no task here. but we try to emphasize a certain internal representation. And this was either memory of landmark memory, so this was the memnet, where the network was encouraged to remember the identity of the last wall encountered, north, south, east, west, or path integration, the POSNet position, where the network was encouraged to remember the actual position of the agent while it was running around. So this was modifying the internal connectivity of the network without any task. Then we trained, using Q-learning, we trained a task, using only the output, the readout of the network. And the task could either be more, let's say, topological in nature, so we scale the arena from different trials, or more metric in nature, so we put obstacles. And when we take these networks, we see that the network that was pushed toward the more landmark, task found a topological solution. It found a solution that did not rely on absolute position, whereas other networks found a more metric-like solution. And if we then use a host of tasks, we can define a topology score and a metric score. And we see that random networks, these are the blue ones, are able to solve it somewhat, but the pre-trained networks are both better, so they move, let's say, in the direction of better performance, but also sort of the position network moves in one direction, the memory network, the landmark, moves in the other direction. And then the question was, what happened here? So how did this come about? And the idea is that pre-training creates dynamical objects. And to find these dynamical objects, we wanted to find what's known slow points in the network. So this is based on work I did with David Cicillo. So if there's a fixed point in dynamics, then this scalar function has a 0 in phase space. And if it's not a true fixed point, but only sort of approximately fixed, then we expect small values of this function. So when we look at these slow points in a random network, we get large numbers. This is the distribution, whereas the trained networks, these pre-trained for a position or memory, have much lower. So they seem to really have slow points and slower ones for the memory network. And now we look at the topology of them, and we see that the position network created a 2D manifold of slow points, whereas the memory network created 4 distinct fixed points or semi-fixed points for the memory. So what we can think of is that A, trained networks have slow points and not fixed points. They create approximate objects. And B, learning shapes the dynamics of the network that then shapes the learning that will take place in the next time. So it's a dynamical process. This is a recurrent network, right? So when you learn something, it changes the dynamics that will affect the learning that goes on next. And to understand how these objects form and how learning appears in a more exact manner, we turn to a more tractable problem. This is work done by Friedrich Schussler. He's a PhD student in the lab in collaboration with Sergej Nostojic, Alexis Dubreuil, and Francesca Mastrogiuseppe. And the idea here is that we are going to train networks using gradient descent without any constraint. And we start with some random connectivity. By definition, after we train it, there's going to be some delta to the connectivity. And we're going to analyze this delta of the connectivity and try to understand whether it has any structure and why. So here's an example. There's a task, the delay discrimination task from the Roma lab, for instance, 2 inputs separated by a delay. And the task is to know whether the second one is larger than the first one. And the network learns this. And now I first want you to concentrate on the dark shading. So this is a case where the initial structure the random part is 0. So there is no initial connectivity. And you see, so this is loss over time, over training time, and it takes it quite a while, but eventually it gets it. If, on the other hand, we do have initial connectivity, and this is the G, so G here is just saying that this is taken from random with G squared over N variance. So this is just the scale of the random connectivity. So if you have initial connectivity, you do this much, much, much faster. But in both cases, the network is able to learn. Now, what was learned? So what we do now is we take only this delta, only the structured part, and we look at the singular value decomposition. And what we see is that it rather quickly drops down to 0. Now it would seem, by the way, both for the 0 connectivity and for the initial connectivity. So it would seem that this is a low rank perturbation. But of course, what is exactly, is it functional? Does it mean anything? So what we do is we do a functional assay. We take these individual ranks and we lesion them one by one to see how it affects function. And you see that if you remove, let's say, rank 6 and above, then nothing really happens. So the first 5, even 4 ranks of the delta W are sufficient. So the solution found by an unconstrained gradient descent is a low rank perturbation to connectivity. And this is true for the Roma task. This is true for the Mante-Sassilo task from the Krishna Shenoy's lab and the Newsom lab. And this is also true for a flip-flop task. In all of them, we see that very few ranks are sufficient to get training. And we see that if we increase the random connectivity, then the training is much, much faster. So initial connectivity accelerates learning, and the learned connectivity is of low rank. To understand this, we move to a linear network. So now we have, instead of having we don't have some phi of x here, but we have just a linear function. So basically, we put some input through vector m, and we read it out through a vector w, and we modify the internal connectivity w using gradient descent. And then the desired output is just to reach some constant value z hat. Okay, so it can be 2, it can be half, whatever we want. And what happens is basically the same. So the performance improves. So learning is faster with initial connectivity. And the singular values, so now this is a bit of a different plot. So this is learning time, and I'm showing you all singular values, but basically one of them is non-zero and the rest are almost 0. So only one singular value increases here. And this is the same, although it's an interesting fact that it's there are some qualitative differences depending on the target value, which we understand qualitatively why this happens. And what we can show in this linear case is if we solve the gradient descent, then the delta W depends on this matrix B, which of course contains W itself. So the dynamics of the network is linear, the dynamics of learning is non-linear, but we can expand that in the learning time. And in the first order, what we get is a rank 1. So this is a vector, and this is another vector. So this is sort of an outer product type. So the result of learning is a rank 1 perturbation, even though we did not constrain it to be so. And we can also understand why learning accelerates with the initial connectivity. This is related to the fact that the low rank part is highly correlated to the initial random connectivity. This is also a recent paper from by Friedrich that analyzes this stuff. Okay. To conclude, complex neural signatures suggest complex models. Surprisingly, a good model for adapting complex systems like the brain could be adapting complex systems like artificial neural networks. But understanding these models requires some new concepts, and both the learning process and the final outcome can be analyzed. So a few structures in this randomness that we can already identify are that the dynamical objects are slow points and not fixed points, approximate, not exact. They act as a prior, or they provide inductive bias for the learning process. And this can also be thought of in the context of online learning and control. There's a recent paper that explores this interplay between learning and dynamics leads to quite interesting things. The low-rank perturbations, at least I was exposed to it as a trained trick in reservoir computing, but it actually arises very naturally, even without a trick. And you can also think of it, why it happens, because every complex system, it can be a brain, it can be a brain area, it can be a cell and a genetic network that has to adapt to its environment. And it does this via a narrow communication channel. So in a sense, whenever a complex system adapts to an environment via a narrow communication channel, this means that effectively you have a low-rank feedback loop. And we also show that this is the case for the topology of genetic networks, where hubs can be thought of as effective feedback loops. So I think this is a rather general theme or a type of structure or simplicity that emerges when studying these complex systems. And that's it. Thank you very much.
Thank you. Yeah, so we have a question from Tom Burns. Could the technique you described in the first study, Grid World, be applied to curriculum learning problems? Also, what would happen if you add more modularity to the RNN?
Okay, so yes, I think it can be used for curriculum learning. We have, let's see if can I switch here to slides? I'm trying to find my, okay. So for curriculum, several answers. First, it can be used as a modular. We tried it in the ICLR paper. We took a modular one that had both the landmark and the position, and we combined them, then it does get somewhat the best of both worlds. As for curriculum learning, there's another work by Doron Haviv and Sasha Rivkind, where we basically we did curriculum learning, and just cut to the main thing here. So what we could see is how the curriculum causes fixed points or slow points to emerge gradually via learning, and why certain curricula are better than others.
I think we'll take one more. I remember, so this is from Dmitry Todorov. I remember in your paper with David, besides the slow points, also the saddles with lots of stable and few unstable dimensions were important. Do they play a role here as well?
Okay, so first of all, slow points are the analog of fixed points. So they can be both. So saddle points are a category of slow points or fixed points. And yes, in general, there was this, let's say the flip-flop task is the let's say canonical example where you have the saddle point in between. So we do, in general, we expect to find them where we have different basins of attraction. Interestingly, in many cases, we fail to find them. So this algorithm is not perfect. There's no guarantee that you will locate all the slow points. And there's by the way, let's see if I find it here. There's another approach by Katz and Regia that also try to find these fixed points using another method, which can be thought of as an intersection of null lines, if you will. But that also doesn't have any guarantees. So I believe that these saddle points are important whenever you have separatrix, basically, but we are not always able to identify them.
Thank you. There's one more question. We'll move that to the Slack channel for this group. If you could please take a look, and we'll move on to our next speaker.
Okay. So thanks a lot.
Thank you. All right. So our next speaker is Lai-Seng Yang, who is a Henry and Lucy Moses Professor of Science at and Neuroscience at NYU. Okay, Lai-Seng.
For some reason, I'm having some problems. Is that okay?
Yeah, we can't see. I don't think we should just see part of your screen. So
I don't know what I can do about it. Or like, how about like it wouldn't let me move you off my screen. So.
Oh, okay. Do you want to stop sharing and then start sharing again? Because we just see, yeah.
Oh, what? How much of it do you see?
It was just a strip.
Okay, so
it worked well before. So
yeah.
Yeah.
Are you seeing it now? I see the entire screen. Are you seeing it now?
No, I just see you, so it's not sharing yet. Yeah.
So okay, let me go back. Okay, I'm sharing screen. Playing.
Okay, there we go. That works.
What? Okay.
Now now we can see it all. All right.
Okay. So hi everyone. So I'd like to talk about today's the structure and dynamics in the model of the macaque visual cortex. So the macaque visual cortex is very much like our own. And on the picture on the right here, you see a picture of the side view of a human brain. So as information enters the eye, going to the retina, and in one synapse, it goes to what's something called a lateral geniculate nucleus, shaped like a knee. And in another synapse, it goes to the back of the head, where the visual cortex is located. So the model that I like to talk about goes from LGN. I skipped the retina part. It goes from LGN to the input layers of the visual cortex, and I will be discussing what is in the magnostream, one of the two main visual pathways into the visual system. Now, this is what I'm going to describe is what's considered to be realistic computational modeling. Now, no model is completely realistic, or it wouldn't be a model at all. So by realistic, what I mean is realistic in the world of models. And this is joint work with Bob Shapley, who's a neuroscientist, experimentalist, and Logan Charaka, a younger fellow. Many of the simulations that you see are done by him. So let me start by giving you some idea of the structure of the network that I'm discussing. On the left, this is not part of the network. This is intended to symbolize the external stimulus that I'm going to present to the network. So the network has three components: the LGN, okay, the lateral geniculate nucleus, and layer 4C alpha, which is the input layer to V1, the primary visual cortex. The input layer interacts with another layer of V1, layer 6, and they talk back and forth. Layer 6 actually also gets some LGN input, but the main input layer is 4C alpha. So now both LGN and 4C alpha are modeled fairly realistically, meaning that we follow neuroanatomy in every way that we can as far as the network neurons and structure goes. Layer 6 is very complicated. It has a lot of very large variety of neuron types and also interacts with other parts of the V1. So we're not able to model that realistically. But in any case, the main object of interest are these two parts, LGN and 4C alpha. Now I cannot really leave out layer 6 because layer 4C alpha gets gets feedback from it, so it has to be there. Okay. So what I'm going to discuss are the structure of LGN and layer 4C alpha, and also the dynamics of neurons in both of these. And in particular, in 4C alpha, I'm going to discuss it on 3 different levels: on the level of individual neurons, on the level of local populations, and also across the cortical surface. Okay, so structure and dynamics of LGN. LGN is a fairly simple object, in the sense, not simple at all, but simple compared to cortex, in the sense that, well, there are 6 different layers. Here's one layer. 2 things that you need to know about it is that one is that it's very sparse. Now, just to shock you a little bit, if you stare at the moon, in the area that covers about a quarter the size of the moon, there are only 10 LGN neurons, on the order of 10 LGN neurons on the magno sheets. So that's very few neurons covering a very large part of the visual area. And the second thing you need to know about it is that there are 2 types of cells, on and off, and here's how they function. So LGNs basically function as linear filters. That's why their dynamics are relatively simple. So the input current reflects fairly well what's in the output. So what goes into the input current in LGN, first there's a background current that's always there, no matter what you do, is there. Okay, and then it's plus or minus some, depending on whether you're an on cell or an off cell, another quantity, which is it's an integral of the light intensity map. So L of X is this light intensity map that describes what the external stimulus that comes in. And there's a spatial kernel, A, and there's a temporal kernel. So the spatial kernel, this is a 2-dimensional object. LGN is really a sheet, a 2-dimensional sheet. So this is the radius of the spatial kernel. So basically it just sees a little spot with a little bit of a center surround effect, but it's fairly weak. This temporal kernel is more interesting. It has a shape like this, which is not the usual shape of a kernel. But what that means is that LGN is a change detector. When you integrate against something like a sine function, what you're doing is taking the derivative of the input signal that's coming in. So for on cells are excited when it goes from dark to light. So each LGN neuron only sees a little tiny little spot in visual space. So it focuses on the luminance of that spot. And when it goes from dark to light, this derivative tells the on LGN cells to spike. They get excited. And the off cells do the opposite. They get excited when it goes from light to dark. And that's basically all that there is to know for the purposes of this talk about LGN. Okay, so again, it's where LGN has, so something I did not stress, I should say, is that LGN has a retinotopic map. Now there's a map of the visual field on our retina, everybody knows that, but that map is passed to LGN and actually onto cortex. So in other words, LGN has, like, there are spatial locations in the sheet will reflect the spatial locations in the real world. But so that's something that LGN has. But on top of that, it really just does this integrating and taking derivatives. There is certainly, it does not recognize edges. So there's no such thing as orientation selectivity or anything like that. It's just very, very simple. 2 kinds of cells. One goes on when it goes from, when things turn from light to dark, and the other one goes on when it goes from dark to light. And that's all the dynamics and structure of LGN. Now, going to the structure of the model cortex. Now, cortex, so I'm mostly talking about input layers of V1. And one of the most salient features of V1 is orientation selectivity, in addition to having this retinotopic map. What orientation selectivity means is that each neuron has a favorite orientation. So it also has a tiny receptive field. And if it sees an edge aligned with that or its favorite orientation, then it's really excited. And when it's orthogonal to it, it doesn't respond at all. And in between, it responds by variable amounts. That's what's meant by orientation selectivity. Now, this is a picture of a real piece of V1. It shows that the neurons preferring the same orientation tend to be grouped together, and these are called orientation domains. So you will see that neurons preferring vertical are colored red in this imaging map, and neurons preferring like horizontal are colored in this blue-green. So you see that they are grouped together. Okay, so that's a real piece of V1 cortex, and here's the model. Okay, so then the model, this is a 9 hypercolumn model. Each square, hypercolumn, each square represents a hypercolumn. There's no such thing. You don't see hypercolumns here. Hypercolumns are just structures that we made up to make it easier for us to talk to groups of orientation domains. So each hypercolumn is further divided into different, these are the regions that correspond to the colored regions. Okay, they are intended, they are regions of intended orientation with intended orientation preferences. So just to give you some idea of the model, which follows real anatomy, each hypercolumn is, this is at about 5 degrees eccentricity, fairly close to when you look directly in front of you. So each hypercolumn individual, it represents about 1/4 of a degree by 1/4 of a degree, like roughly the size of 1/4 of the moon, okay, when you look up at the sky. And in this layer alone, there are 4,000 neurons in each hypercolumn, 3,000 excitatory and 1,000 inhibitory. That's just one hypercolumn, one layer. So you can see that these 3,000 and 1,000 here, as opposed to LGN, which has only about 10. It's a very, very big difference in number. And so in this model, the neurons are modeled by leaky integrated and firing neurons. And here's the connectivity. So I want to stress that to the degree that we can, everything we try to do is models realistic densities in real cortex and realistic length scales. But of course, hypercolumns are not square, that we made up, so that otherwise it's very, very hard to communicate with the computer. Okay? So, so this is an E neuron, and you're looking at randomly drawn, pretty much isotropic groups of neurons that are connected to it at connection densities that are that mimic real cortex, and is fairly it's quite sparse, as you can see. Whereas this is E to I, so an I neuron sitting in the middle and E neurons like that, and on the side, the most of the neurons in this layer are basket cells. They have very short axons. That's why it's the cloud is smaller. So this is mostly the structure of the layer 4C alpha. Layer 6 is a layer that's very similar to this, but let me focus on 4C alpha. So this is pretty much the structure of 4C alpha. So now the next thing I have to talk about is how is LGN connected to 4C alpha. So in particular, I need to answer the question, how is orientation selectivity conferred on V1 neurons? As I said before, LGN has no orientation selectivity. It only goes light, dark, light, dark. It only cares about changes in luminance. So there is no way you can see edges from they don't know anything about edges at all. Whereas V1 neurons, they all have favored orientations. And yet there is nothing between LGN and V1. So how did this orientation selectivity come about? Okay. Well, the answer was suggested to us or provided for us, which however you see it, by Hubo and Wiesel. So in this scheme, cortex is divided into regions of intended orientation preference, as I've shown you before. So these are the bars indicate the intended orientation preference. And the task in building the model is to each neuron in V1, we're going to assign a handful of LGN cells, a number of LGN cells that are supposed to be spatially aligned in 2 or 3 stripes of on and off. This is what Hubo and Wiesel showed us, and I'm going to show you what I mean by that. But first, a word about the number. Up until up until fairly recently, people used to think that the number of LGN cells connected to a single V1 neuron, it's like in the dozens maybe. And it was only found not so long ago, mostly from the experimental result of Angelucci and also the sparseness of LGN cells also, that is really only very few. It's only a handful. Okay, so here's what I mean by rows of on and off. So these unfilled and filled circles represent on and off LGN cells. And this is a row of on, and this is a row of off. So Hubo and Wiesel says that we get cortex gets orientation selectivity by connecting to these rows of on and off cells. So now the moment that we start to do this, there's a combinatorial geometric problem that comes up, is that, well, since you can't reach very far, so like roughly this many, how many on-off stripes can one draw from a set of about 15 to 20 LGN cells, half on, half off? You can see that it's really not so easy to draw. I can draw a horizontal one easily, but there are not that many angles that you can draw from. It's very, very discrete. And how do you represent orientations around the clock? And there's another issue, is that the distance between the on and off have to respect certain laws about that show up in the spatial frequency preference. Okay, so the answer is it can be done, but barely. And here are some example stripes that we have. So for example, here is this is a stripe, an on stripe, an off stripe, and an on stripe. So this is a 3-stripe cell with 3 of the stripes according to Hubo and Wiesel. Here's one with 2, and off, and then on, and so on. So maybe I should say a word about how why are these stripes? How do they confer orientation? So take an example of this 2 off, 2 on one. If you have a grading that's vertically aligned, I am claiming that a V1 cell that's connected to these 4 LGN cells will be very sensitive to vertical edges. And that's because when a vertical grading comes, imagine it's coming from right to left like this. Oops. Okay, it's coming from right to left like this. If the spatial frequency of the grading matches this distance between the on and off row, there will come a time when the on cells are very happy because they see white, and the off cells are very happy. So all 4 cells are very happy, and they spike together. And when all of the LGN cells spike together, it's very powerful. It sends a very, very strong signal to cortex. Now imagine that you have a grading that's aligned some other way, coming say from some angle like this, okay, so some different angle. You will excite 1 or 2, maybe 3, usually 1 or 2 at a time. You will excite them eventually, but they are not excited simultaneously. And so the message sent to cortex is much weaker. And this is what Hubo and Wiesel suggested, and we verified in the model. Okay, so this is all that I'm going to tell you about the structure, except that I haven't said anything about layer 6. Layer 6 is the firing rate in the model is indexed to layer 4. So layer 6 is another sheet. Think of it as sitting below layer 4. So it has the same kind of spatial locations. And so what we do is to just make layer 6. Layer 6 is very hard to model because it's very, very complicated. So all that we do is to have it feedback something which is indexed to layer 4. If layer 4 firing is high, it sends back a feedback that's high, and layer 4 slow is low in the same location at the same time. Okay. So this completes the description network. And next I'm going to talk about dynamics. But before I get into that, I want to say that once the parameters are chosen, I'm not allowed, the modeler, in other words, me, I'm not allowed to touch the model. The model is going to, so once this input goes in, the model will compute on its own. LGN integrates all these things, sends to V1. The neurons here interact with layer 6, and layer 6 comes back like that. The model is going to compute on its own, and it's going to follow the mechanics of neuronal interaction in real cortex as far as we know. Okay. So about dynamics, first I want to talk about the dynamics of individual neurons versus local populations. So, and both of these things have been shown to, so every step of the way in the building of this model, we have tried very hard to make sure that everything matches data. So the number of sets of data that are matched, I don't really know. It's many dozens. It's hard to count whether they're the same or different. Okay. So first, for individual neurons, there's something very, very well known, that you take an individual neuron and you measure the time intervals that passes between spikes, you will see a distribution that looks like this, interspike intervals. It looks very much like an exponential distribution. It need not be exponential, but it just has a very long tail. But if it was an exponential distribution, it would really basically mean that the next spike is is going to come at a fairly random times. It's like a Poisson process, right? And this is when it's driven by a grading at 4 Hertz. So you can see that roughly 200, 250 milliseconds later, there's another little bump. But in short, if you look at a single neuron, the spike firing times of a single neuron appear to be very, very random. In fact, one way that one could rationalize that is that each neuron receives a fluctuating current because it receives a current from many, many other neurons. So the sum of which is a fluctuating object function that produces a random walk-like behavior with a drift up towards threshold when you are driving it. And so ISI can be modeled as a first passage time from reset to threshold as inverse Gaussians, and they have shapes that look like this. Now, individually, this is what neurons do, but when you put them all together, something else happens. Okay? In the model, they produce a gamma rhythm, which I'm sure most of you know about. So this is a raster in the time in the horizontal axis, and neuron number. Blue is I neuron, red is excitatory neuron. The ones that fired further down below, they fire more because they're complex cells. Okay, so you can see that the spike firing is very far from homogeneous. It produces a rhythm, and I want to emphasize that no organization, there was nothing that I did to cause this to happen. The neurons are certainly not grouped in any way. They are kind of randomly, they're randomly picked, and yet they self-organize into this kind of spiking pattern with the frequency at a gamma band. Okay, and this is an entirely emergent behavior. Okay, so just to say a little bit, so gamma band rhythm is detected, as you all know, in many parts of the brain, and it also shows up in models. You take a group of neurons, you randomly connect them together, and a gamma rhythm will, if you respect the physical time constants, I should say, and respect the fact that E and I are in good balance, then a rhythm will appear, and it's almost hard to get rid of. It's just there. Okay, so this is just a picture from the previous slide. In case you think that these are population spikes, here's the percentage of neurons that are firing at any one point in time. As you can see, most of the time it's less than 10%. Occasionally you get a big one like 20, but is extremely far from synchronous firing. Okay? And if you look carefully, it's actually not the same ones that form that group together to fire also. So neuroscientists have done a lot of computation of the power spectral density. Also, one of the reasons for looking at this is that you look at this, they call this a rhythm, but it's not really exactly a periodic, is it? Some are really wide, some are narrow and get messed up, and some are kind of come on stronger, and some are fainter. Okay, so this is described by this power spectral density, and what this is that n is the total number of neurons. So you look at the time and divide it into a lot of different time bins, and then you look at UN is the instantaneous firing rate, the fractional neurons that's firing, rather not instantaneous firing rate, sorry, is the fractional neurons that's firing in the nth bin. And so what you get is really a signal, an infinitely long signal, which is very far from periodic, but you pretend that it is, and you look at the first like 200 milliseconds or so, and you compute its Fourier coefficients. And then you time shift that time interval by a little bit, and here's what you get. So this is a fairly realistic picture in the sense that you almost cannot tell it apart from the real experimental data. So this is time. So here's very long. This is 1 second. Between here and here is already 1 second. This is the frequency. And you can see that the power is mostly concentrated around like here, maybe about 60 Hertz, which is in the gamma domain. But it wanders. So this is the wandering frequencies. This is a character of gamma signal, and it's completely, entirely emergent behavior. It's 60 for a while. It could disappear. It could drop to 50. It sometimes goes up to 80, 90. It just wanders around. That's a caricature of it. Okay, and so there are different recurrent excitation inhibition is what is it's an explanation for it. Since I'm running short in time, I'm not going to explain that, as it takes a little bit of time to explain. Okay, so what I'd like to show you are some pictures of some, this is what the model cortex looks like when it's stimulated. So look at first this picture. This is when you're passing a vertical grading through. Now, here's the key, the translation. If you go to the model cortex, these are the regions that are colored purple, are the regions that are supposed to be intended to be sensitive to vertical grading. They are wired, meaning they are wired as such. Of course, you don't wire everything perfectly, but they are predominantly wired to prefer vertical. And indeed, when you present a vertical grading, these are the parts that light up. Lighting up means that it's firing high. This means dark. This is E and this is I, and you can see that the eye neurons are more broadly tuned, so it's got a much bigger footprint in this picture. So if you go to horizontal 90 degree, you will see that these are the diamonds, and they correspond to these. And what if I go to 45 degree? This is a 45 degree grading. If you look here, you don't see 45 degrees, but you see 30 and 60 degrees. So what a 45 degree one responds to is some combination of 30 and 60 degrees, so you can see the correspondence between this intended wiring and the actual response of the neuron. So just to focus on the central hypercolumn, you will, people talk about this pinwheel structure. You will see as you go along 0, 25, 45, 60, like that, okay? And to remind you again, this is the connectivity. It does not respect this, it crosses the orientation boundaries. It is fairly isotropic. Okay, so this is I have a movie that I like to show you, and this is exactly what you saw before, except that what I showed you earlier was average over 1 second, and this is happening in real time. Well, not in real time, I've slowed it down or you wouldn't be able to see it. So here is the time clock in milliseconds. It's gonna be, it's moving. And you can see that I'm starting with a vertical grading, and these are the diamonds that correspond to the vertical grading. So I want to emphasize that the model is really computing on its own, and it's following the way the brain computes. In other words, it's alive, okay? And so here it goes. Okay, so this is a vertical grading going. In a moment, it's going to change to another grading, change to a 25 and 22.5 degree grading. So you notice that a different part of the cortex is lit up. Okay, in a moment, it's going to change again to a 45. This is the horizontal grading. Okay. Okay, so so much for the movie and summary and conclusion. Okay, so we have built a structurally realistic model of the Macaque V1 cortex. And by structurally realistic, I mean following the anatomy of the real cortex on the level of neurons and circuits. Because we did not model what goes on inside neurons or intracellular properties are not modeled, but on the level of neurons and circuits is as realistic as we could do it. And with biological rules of dynamical interaction, meaning that LGN are change detectors, we model them as such. The LGN to V1 connection, we follow Humo and Wiesel. The dynamical interaction, the length of axons, and so on are intended to model real cortex. And what we found was that in cortical dynamics was that it's really quite different if you look locally versus looking globally. If you look locally, it mostly depends on an EI balance. You see if there's a balance of the excitatory and inhibitory neurons, as well as the push and pull that produces the gamma rhythm. But as you look across the cortical surface, then it's a different story. It depends on many, many things. First of all, it follows the stimulus. It better follow the stimulus or we wouldn't be seeing anything. But it's also a complicated balance between the feed-forward input, the lateral contribution, and feedback. And here, both structure and dynamics of feed-forward, lateral, and feedback all count. So the feed-forward would have to be what LGN gives it. The lateral in this layer, there are no long-range connections, but otherwise it matters whether or not there are long-range connections, and it matters how much feedback. Mathematically, this can be described as a non-equilibrium steady state. It's a concept borrowed from statistical physics, And there's a fair amount of mathematics and physics associated with that. So this is all that I wanted to say. And my team, again, is Logan Charaka, the fellow on the left, Bob Shapley, neuroscientist, and myself, and thank you to the Schwartz Foundation and to NSF. And thanks for listening.
Thanks, Lai-Seng. So if there are questions, please type them in the Q&A box. In the meantime, I had a question. So what happens if you present, so this is kind of a two-part question. What happens if you present more complex stimuli? And how could you, and as a result, to turn this around, how would you verify this model accurately captures cortical responses?
Okay, so the structure of this model is certainly not built to only replicate the dynamics in response to gradings. Okay, but it's very small. It only has 9 hypercolumns, which means that you cannot. It only sees an area that's roughly 0.75 times 0.75 degrees. So first of all, I will have to extend it spatially in order to see more interesting objects. But as far as the structures go, this is all there is, basically. So if this model is supposed to capture everything in this tiny piece of the visual cortex, independent of the nature of the stimulus.
And so what would happen but it excludes top-down projections, right? So that could modulate those responses.
Yeah, that's right. If you, in trying to model the visual cortex, I think that we have a fairly good chance of success modeling the volley of information that's coming in. Top-down things are very hard to model. So for example, V1 talks to V2, to MT, to all these things, and MT and these other V4, V5, these areas, they talk to other regions of the brain and it bounces back. We're not in a position to model, certainly not a detailed, accurate modeling of this kind to deal with any of those. So what we are really trying to capture here is the input volley of information. So if you've thought about it and decided, well, you know, yes, it modulates what you see, but still it's dominated by the input information in these layers.
Thank you. And so this is all the time we have. So thank you to Lai-Seng and to all the other speakers. As I mentioned before, we have a Slack channel devoted to this particular session, parallel session. I just checked that there's most of the questions that were posed and not answered directly during this time have been answered on the Slack channel. If you have further questions, this is also a good place to ask the speakers again. So thanks again. And I think we will reconvene in about 25 minutes, or rather there's going to be another talk in 25 minutes. And that's it.
Yes.
Goodbye,
so,
everyone.
Yeah, so we will reconvene at 17:30. There will be a coffee break now. And yes, so there will be a session on the mathematics of deep neural networks in channel 2 and a session on neural field equations and spatially extended systems in channel 1. Okay, so hope to see many of you in the next sessions. So thanks again, all the speakers, also from my side.
Thank you as well for inviting us.
Thank you.
Julian.